Regulation of gene expression. A gene which encodes a product required for the maintenance of basic cellular processes is called a housekeeping gene. The gene is also called a constitutive gene, as it is an unregulated gene whose expression is uninterrupted. There are some genes which regulate the expression of other genes and are called regulator genes. Gene regulation is brought forward by François Jacob and Jacques Monod in 1961 in E. coli. They put forth the Operon hypothesis. Operon hypothesis says that gene regulation in prokaryotes and bacteriophages involve structural genes, the operator, the promoter, the regulator genes, repressor proteins, and an inducer. The expression of genes is regulated by the metabolic, physiological, or environmental conditions. The Lac Operon. A genetic unit comprising of one or more structural genes, an adjacent operator-promoter region that controls the transcriptional activity, is called operon. Operator and promoter are cis-acting elements which are located on the same piece of DNA. These are situated before the structural genes. The molecules which attach themselves at sides away in the piece of DNA of structural genes constitute transacting elements. Components of Lac Operon. Structural genes. Z, Y, and A are three structural genes of lactose operon in E. coli. Z gene codes for enzyme beta-galactosidase, which hydrolyzes lactose into glucose and galactose. Y gene codes for permease. It affects the lactose uptake from the medium being present on bacterial cell membrane. A gene codes for the enzyme transacetylase, whose function is not clearly known. These genes are sequentially transcribed as a single mRNA from a single promoter and are called structural genes. The mRNA synthesized is called polycistronic mRNA. Last cistron is provided with termination codons. Operator region. Operator is situated upstream to structural genes. When it is on, the structural genes transcribe mRNA. When it is off, there is no functioning of structural genes. Enzyme RNA polymerase is responsible for transcription of mRNA. It binds to the promoter region and moves along the operator region and structural genes. Repressor protein produced by the regulator gene binds at operator to regulate the synthesis of enzymes. Promoter region. Promoter region is the actual site of initiation of transcription. It is upstream next to operator region. RNA polymerase, the enzyme catalyzing mRNA transcription, binds to the promoter region and moves along operator and structural genes. Regulator gene and repressor protein. Regulator gene synthesizes mRNA coding for a protein called repressor. It is an allosteric molecule which changes its conformation and chemical activity if another molecule binds to it. In the absence of lactose, repressor binds to the operator and blocks the path of RNA polymerase and prevents the expression of structural genes. Inducer. Inducer binds to the repressor and changes its conformation such that it cannot bind to the operator. Therefore, the RNA polymerase initiates the expression of structural gene to produce respective enzymes. Few molecules of lactose in the cytoplasm of E. coli are converted into allolactose and isomer of lactose. These molecules induce the expression of operon by binding to the repressor. These are called as inducers. Such operons are called as inducible operons. Regulation of Lac Operon. E. coli contains a few molecules of beta-galactosidase enzyme. Beta-galactosidase in the cytoplasm metabolizes lactose into allolactose, an inducer. Allolactose binds to repressor protein, which detaches from the operator. RNA polymerase initiates the transcription of structural genes to synthesize polycistronic mRNA. Permease synthesized from mRNA helps in rapid uptake of lactose. Beta-galactosidase molecules in the cytoplasm metabolize lactose into galactose and glucose. Constitutive mutants confirm Lac model of regulation. If mutations occur in regulator gene or operator region, the regulator gene is the constitutive gene.